Bismillahirrahmanirrahim. Today we are going to start a very important lecture that is about antibodies. Antibodies, they are also called as immunoglobulins. These antibodies, they are globulin proteins and they make up about 20% of the total plasma proteins. These antibodies, they have 5 important classes which are IgG, IgM, IgA, IgD and IgE. Here the term Ig, it stands for immunoglobulin. So immunoglobulin G, M, A, D and immunoglobulin E. So we divide these immunoglobulins in 5 different classes or 5 different groups on the basis of their heavy chains. Most important, when we talk about the important functions or the most important function of the antibodies, that is they neutralize the viruses, they help in opsonization of the bacteria, they also help in neutralization of the various toxins, they can cause the activation of the complement cascade and prevention of the microbial attachment to the mucosal surface. That is a very important function of the immunoglobulin A molecule. So these are the important function you have to note down: neutralization of the toxins and the viruses, activation of the complement cascade, opsonization of the bacteria, and prevent the mucosal attachment of the microorganisms. Structure of immunoglobulin molecule or structure of antibody. The structure of simplest immunoglobulin molecule. Immunoglobulin molecule, it is made up of light chains and heavy chains. There are total 4 polypeptide chains, 2 light chains and 2 heavy chains. And these light chain and heavy chain in the form of simplest immunoglobulin molecule, they are arranged in a Y-shaped structure. These light chains and heavy chains, they are linked together or they are held together by the disulfide bonds. Each light chain, that is denoted as L, and each heavy chain, that is denoted as H, they contain variable regions and constant regions. Variable regions, they are denoted by V, while constant regions, they are denoted by letter C. So this is the structure of immunoglobulin molecule which I discussed in the previous slide. As you can see, each immunoglobulin molecule, it consists of 2 light chains and heavy chains. The light chains, that they are denoted by yellow color, while the heavy chain, they are denoted by somewhat red or pink color. And these light chains and heavy chains, they are held together by disulfide bonds. The disulfide linkages, they are shown clearly in this picture. There are 3 or 4 important points to note in this picture. As you can see that each of these chain, each light chain and each heavy chain, it is divided into various segments. It is divided into variable region and constant region. And variable region, that is denoted by letter V, while constant region, that is denoted by letter C. So the variable and the constant regions, they are present in both the chains. Their number, it differs. If we talk about the light chain, it consists of 1 variable region and 1 constant region, and you can see clearly in the yellowish portion, that is denoted as VL and CL. VL stands for the variable region of the light chain and CL, it stands for the constant region of the light chain. While on the other hand, if you look at the heavy chain, it consists of 1 variable region denoted as VH and 3 constant regions or 3 constant domains. And each of the constant domain, that is further denoted as CH1, CH2 or CH3. The antigen binding site or the region where the antigen it binds, that is present in the variable region of the immunoglobulin molecule. So the variable region of both the light chain and the heavy chain, they are responsible for the antigen binding, while the constant region, they perform some other functions. For example, if we talk about the second constant domain of the heavy chain, that is denoted as CH2, it serves as a site for binding and activation of the complement. So complement cascade, it binds to this CH2 domain, the constant second domain of the heavy chain. One important thing to note about the that is related to these variable regions is the hypervariable regions. The variable regions of both the light chain and heavy chains, they have they have an amino acid sequence that comprises of 3 amino acids, and these 3 amino acids, they are present at the terminal end, and uh, these amino acids, they result in the formation of a hypervariable region. So this is the region of the hypervariable region. It is the region of the antigen binding site that comprises of 5 to 10 amino acid sequences, and it is here at this hypervariable region that the antigen binding it occurs. So the light chains, when we talk about the light chain, they are of two types. One is kappa and second is lambda. And when we talk about the heavy chains, they differ. Heavy chain, they differ with respect to the type of immunoglobulin molecule. They may be alpha, they may be gamma, delta, and epsilon or mu. There are two important points to note about these heavy chains and the light chains. At a single time, only one type of the light chain will be present, either kappa light chain or the lambda light chain. While when we talk about the heavy chains, in a single immunoglobulin molecule, only one type of the heavy chain will be seen. It may be alpha, gamma, delta, mu, or epsilon. There will be no combination of the two light chains or the heavy chains. So at a single time, there will be only one type of the light chain and one type of the heavy chain that is seen. If you look at the right side of this picture, you can see that two terms are written: FAB, FAB, and FC. So these are the two important fragments, the FAB fragments and the FC fragment. The FAB fragment, it contains the antigen binding sites, while the FC fragment, that is involved in the complement fixation, complement attachment, and attachment to the various other cells and some other biological properties. Please note another important point about the light chain. Likewise, if like I told you about the constant domain of the heavy chain, that they perform few important biological functions like complement fixation, complement binding, and activation. Do this constant domain of light chain, it performs some function? The answer is that this constant domain of light chain, it performs no biological function. So this is an important MCQ. The constant region of light chain, it performs no biological function, while constant domain, or the second constant domain of the heavy chain, it serves as a site for complement binding and activation. This slide, it shows the previous previous information which I shared in the last slide. All of the things written here, they are told in the previous slide. Yes, there is one thing that was missing in the previous slide, and that is the forces that are involved in the antigen and antibody binding. Please remember, there are two types of the forces that are involved in binding of antigen and antibody. They are the electrostatic forces and the van der Waals forces. While if we talk about the forces or the bonds that keep the light chain and heavy chains together, they are the disulfide bond. Well, forces involved in antigen antibody reaction or antigen and antibody binding, they are the electrostatic forces and the van der Waals forces. Van der Waals forces, electrostatic forces, you people have a very clear view in FSC, or you people may have studied in the second year MBBS. Moving on to the individual molecules, individual immunoglobulin molecule. First of all is the immunoglobulin G molecule. Immunoglobulin G molecule, it comprises of two light chains and two heavy chains. Again, they are held together by disulfide bonds. This immunoglobulin G, it has four subclasses denoted as IgG1, IgG2, IgG3 and IgG4. There are few important points to note about immunoglobulin G is that it is the predominant antibody molecule, predominant immunoglobulin molecule that is present in the serum or in the blood, and it makes up to 65% of the total immunoglobulin molecule. This immunoglobulin G, it can cross the placenta, so there is transplacental movement of immunoglobulin G. Immunoglobulin G molecule, it helps in the activation of the complement, and complement it binds to the second constant domain of the heavy chain. Which function will be performed by the immunoglobulin molecule? It depends upon the various sugar moieties that are attached to the constant domain. For example, if immunoglobulin G molecule, it has N-acetyl glucosamine, it will be pro-inflammatory in nature. It will help in binding and activation of the complement. While on the other hand, if immunoglobulin G molecule, it has a sialic acid side chain or sialic acid sugar attached to it, then it becomes anti-inflammatory. So the type of the sugar attached to immunoglobulin G molecule, it will define the function of immunoglobulin, whether it is anti-inflammatory or whether it will be pro-inflammatory. Next is immunoglobulin A molecule, the main immunoglobulin that is present in the secretions such as saliva, tear, the intestinal secretion, and it is also present in the genital tract secretion. This immunoglobulin A molecule, it gives the mucosal protection. It prevents the binding of the microbes, it prevents the binding of bacteria to the mucosal surfaces. Immunoglobulin A molecule, it comprises of two light chains and two heavy chains. There is a joining component and there is a secretory component that is also present in the immunoglobulin A molecule. Immunoglobulin M, the most important antibody of the primary response. It is present on the surface of all the B cells. This immunoglobulin M molecule, it comprises of two light chains and two heavy chains along with the joining chain. Immunoglobulin M molecule, it is mainly present in the form of pentamer, or it can also be seen in the form of dimer. But the main structure, the main formation of this immunoglobulin M is, it is a pentamer. So it will be having 10 antigen binding sites. When there are 5 immunoglobulin molecule, it means there will be 10 antigen binding sites. So this is the reason this is the most efficient and the most important immunoglobulin molecule in primary response and in activation of the complement and various other reaction where antigen antibody involvement that occurs. Immunoglobulin E molecule, two very important functions that are related to immunoglobulin E, that it is involved in the immediate hypersensitivity reaction, and secondly that this immunoglobulin E molecule, it gives protection against the parasitic infection. Immunoglobulin E molecule, it binds to mast cells and basophil, resulting in degranulation of the mast cells and the basophil. Normally in the blood, the level of immunoglobulin E, it is very negligible. It is present in the trace amount. Immunoglobulin D molecule. When we talk about immunoglobulin D molecule, this antibody, this immunoglobulin, it has no known antibody function but can serve as an antigen receptor. It is present on the surface of many B cells or the B lymphocyte. Again, its level is negligible in the serum. So this is the last slide of today's lecture. The purpose of this slide is to show you the pentamer form of the immunoglobulin M molecule and the basic structure of the immunoglobulin A molecule. If you look at the immunoglobulin A molecule, there are two wide-shaped immunoglobulin molecules, and in the central portion they are donated by a J chain. This is a J chain that is uniting the two immunoglobulin molecule. And if you look at the picture B, that shows the secretory component or the secretory form of this immunoglobulin A molecule. So the basic structure, that is the same two immunoglobulins that are joined together by a J chain, and this J chain that is red in color, and there is a arc shaped secretory component that is present on the surface of both of these IgA molecules. So this is the secretory form of this immunoglobulin A molecule. And if you look at the lower picture, the picture C, which shows the pentamer form of the immunoglobulin M molecule, so you can see there are 5 immunoglobulin M molecules, and the 5 immunoglobulin M molecule, again they are united by a J chain, resulting in the formation of a pentamer. So there are 10 antigen binding sites, 5 immunoglobulin molecules and 10 antigen binding sites, and this is the reason this is the most potent antibody. Immunoglobulin M is in the primary response, in the activation of the complement, or in other reaction where antigen antibody binding occur. So this was a very important lecture about the antibody, the basic structure, the types of the antibodies. इसमें से काफ़ी सारे MCQs निकल आते हैं. Structure of antibody, that is usually asked as an SCQ, as a SCQ. It can be asked to, you can be asked to draw and label the structure of immunoglobulin molecule. You can be asked to elaborate or to briefly describe the structure of immunoglobulin. So a very important lecture as far as immunology is concerned. So now we'll meet in the next lecture. Till then, Allah Hafiz.